control through graphs, but then do at advanced speed. So I think it was to go ahead and add it
Hello, bonjour, merci, Minaklate Nade. Welcome to Nahanni National Park Preserve. My name's Sarah. I'm an ecologist for the park.
My name's AJ. I'm a technician working for Fisheries and Oceans Canada.
So we're out here on the very upper end of Wrigley Creek looking for bull trout.
Bull trout are the fish we all care about. Important cold water species that's an important indicator for lots of other fish species in the park.
We use them to help us monitor what's going on in the park, if there's any changes happening to our ecosystems.
We're using several different methods to catch fish on this project. Electrofishing is one of them. So I got the magic wand here and my Ghostbusters backpack.
And then I'm gonna be following behind AJ, and I'm kind of like the goalie. Gotta catch those fish as they come downstream towards me.
There we go. So we've captured our first bull trout for the day.
Woohoo!
Yeah. So we got a nice size one here. 22.
The other technique that we're using is something a little bit more recently developed called environmental DNA, or eDNA. We don't need to see or catch the fish to know that they're there. We can just take a water sample and then ship them off to the Fisheries and Oceans Genetics Lab in Winnipeg. And they process the samples and tell us if there were bull trout in this stream or not.
One of the best parts of this job is just getting to sit in the pools to cool down on a hot day sampling.
Absolutely, and I reckon we deserve it. It's been like a busy, very full-on week, but it's been a great time. So it's the end of the day, like there's the helicopter coming in to pick us up. Another successful day, some more bull trout.
One of the things people always ask me up north is what's the light like up at night? Well, here we are looking out at the sunset, and it's 12:30 in the morning.
So it's our last field morning today. Myself and one DFO will be sticking around camp to do some sample processing. Let's go get the day started.
Sarah, come check it out. Our results are in. Check it out. So,
What am I looking at?
looks like the reds, our controls were working. Didn't see much with the greens. Nothing in the next 3 samples. Woo! Bull trout.
Really cool to see that happening just like in the field while we're sitting in this cabin. Like,
Absolutely.
not having to wait, as long.